Hello, my name is Eric, and in this work, we're trying to understand something about how host physiology, so by host, I mean the bacteria, how they control the infections of the phages. So I'm going to show again the lipid cycle. So here we focus on T7, and it has all these very textbook injection, expression, and lysis, assembly and lysis steps. And we know that for a fact that all these steps, they have some dependencies on the physiologies of the host, right? There's targets, there's the early transcription, which depends on the polymerase. There's the protein expression that depends on the ribosome, and then there's the packaging that depends on the ATP level, and then eventually there's lysis that depends on the cell wall. So we're trying to understand which physiological factors actually are very critical and also in different contexts, right? So in Pana's presentation, she talked about dormant states, and here I'm going to be talking about the exponential phase. And also we're trying to understand how they impact the lysis or the successful phage infections, and eventually how can we use this knowledge to our advantage to develop better phage applications. Cool. So before that, I'm going to show you an example that we did with antibiotics. It's a simpler model. It just targets a specific thing. So in this example, is translational inhibiting. So the target is ribosome, and then you have probably some influx and outflux of the drug into the cell as well. So you have this very simple model that has an external concentration, inside concentration, binding, unbinding, and also the ribosome is replicating itself, and the cell growth that's diluting the ribosome. And from this kind of treatment, we already see a good heterogeneity in the population. So if you just dispose the isogenic population of the pad and vary the concentration of the antibiotics, at some sublethal concentration, you start seeing this heterogeneous response, right? Some cell grow into large colonies, some cell grow into small colonies, and some will not grow at all, but be able to recover, or some are not able to. So we try to understand this using the mother machine again. So I'm going to show you the image, which kind of just, video, which illustrates how the response is heterogeneous. And now we switch to antibiotics, and you can see how cells kind of start reacting to it. Some will start to live, some are still growing slowly, whereas some kind of just gets down. But when you remove antibiotics, some of them are able to grow back. So with this kind of platform, we can maintain the environment to a very well-defined and homogeneous state. And then we can use the single cell resolution and time resolved imaging to actually probe what's the dynamic and what is the dependencies in there. And also we do observe that for the efflux, so which pumps out the antibiotics, there is some heterogeneity, actually age-dependent heterogeneity inside the population. And also we see some heterogeneity in the ribosome distribution as well. Cool. So what you can do with it is you can analyze the data, you can segment them and track them, and then what you will get is this quantified cell growth traces, right? The cell is growing and dividing. You hit it with antibiotics. For this particular strain, they just grow slower, and then if you recover it, it will just go back to its fast growth again. But if you knock out the efflux pump, which pumps out the antibiotic, it's just not going to have a great time, and then it's not going to be able to recover as well. So we see this, and we can quantify that growth distribution as well. And we can kind of observe the heterogeneous response. So we also see that for the cells with different efflux pump abundance, they are having this slightly different growth under the sublethal concentration treatments. And similarly, for the ribosome as well, if the cell has different ribosome concentrations, they're going to have different fates during the treatment. Some are just going to last, some are just going to persist. It's going to be tolerant and keep going. So then our next question is, can we apply this to phage infections? So this is Charlie's work again. You've probably seen this, where we have the T7 engineered by our collaborator in Fusco's lab. So the cell will expresses the phage genome and start expressing the yellow fluorescence protein when it gets infected. And then it's going to burst, and then that's going to spread and infect the entire micro channels inside these mother machine trenches. So what we're trying to understand then is to adapt the system and use that to understand what kind of physiological factors really are important here. So the first thing we saw is that the phage gene expression actually has a very nice linear, not very nice linear, but it actually scales with the length of the bacteria. So the larger the cell is, actually, the more the phage genome gets expressed. So actually, this can be explained by a very simple model where you just when the phage gets injected, obviously the DNA is going to get replicated, and then it's going to get transcribed, and it's going to get translated, and there you see your signal, right? If you actually look into this model, what it's going to demonstrate to you is it has two phases of phage gene expression. The first phase is where it starts to kick in, and the expression level is dominated by the mRNA abundance in your cell. Then very quickly, that is going to exponentially go into a saturated state where every mRNA just occupies the ribosome inside the bacteria. And then your expression will get determined by this linear regime, where the linear rate is your lambda2, which is the translational capacity. So now you've saturated your translational capacity in your host. And we can fit this to various lots of data, which we do observe these two regimes of phage gene expression. And what we see is what determines the maximum phage genome expression is actually the translation capacity of the host in this context, in the exponential growing cell in T7. So we would obviously want to understand more about this, and the translational capacity has to have something to do with the ribosome. Right? And if you look at the cell growth rate, well, the ribosome concentration is probably going to fluctuate, yes, but it's probably also tightly regulated. But what we do see is this ribosome abundance that actually goes with the cell cycle, which means you have a phage that infects somewhere around here when the cell is just about to divide, and it inhibits that division. You're going to have a lot of burst size compared with a phage infecting a cell that is just newborn. And we did validate this with experiment. We did see that actually it is the total number of ribosomes that scales with the phage gene expression. So what this means is we can now perturb the cell cycle. We can use some lectime to stop the division and make the cell basically a larger bag of ribosomes. What that means is you're going to have a larger size, a larger lambda2, which means your gene expression is going to have a faster linear regime in your second phase. And this did show us an extended relationship towards the right here, where you get a lot more phage gene expression with more ribosome inside the cell. Right, so far I've been talking about the phage gene expressions. What I haven't talked about in our assay is our ability to track also the phage landing and also injection. I'm not sure whether you saw that clearly. I can play that again. Yeah, so you're gonna see a spot landing somewhere around here. That is the stained phage with cytox orange. And you're going to see that signal dimming because that's the phage injecting into the bacteria. And then very soon that bacteria is going to stop growth and then start expressing the yellow fluorescence protein. And then it's going to lyse. So we plan to also use this setup to look at all the steps in the infection cycles and how that depends on the heterogeneity. And also this will give us a principle, like a mechanistic insight into like what kind of antibiotic we should combine with phages, right? What kind of antibiotic, what kind of phages, what kind of bacteria, what kind of strain, and also what kind of dosage. And then also with this like small volume manipulation and nice precise control in the microfluidic device, and use that to do rapid screening and then also characterization of phage and antibiotic complex. And with that, I would just thank you.